Hello, everybody, and welcome. My name is Aaron Essery, viticulture and enology at Oklahoma State University. Today, we have Dr. Patrick O'Brien from Texas A&M University, assistant professor and extension viticulture specialist, to talk to us about establishing and maintaining grapevine perennial structures. So, Dr. O'Brien, the floor is yours.
Aaron. So as Aaron just mentioned, I'm Pat O'Brien, and I am the assistant professor and extension viticulture specialist for the High Plains and West Texas with Texas A&M AgriLife Extension Service. So I've been here in Texas for just coming up 2 years now, but before that, I had spent the prior 7 years living in Australia and conducting research over there. And so most of the work I'm going to be going over today was actually conducted over there in Australia relating to the establishment and maintenance of grapevine perennial structures for long-term vineyard health and productivity. Here we go. So grapevines, when grown under the right conditions, can display impressive longevity, and the fruit and resultant wine of older vineyards is typically perceived as having higher potential quality. In spite of this, however, it is not unusual to see vineyards undergo major reworking or replanting after only a few decades, either to be replaced with other cultivars or due to a decline in production relative to the crop value. A number of factors may on their own or in a cumulative manner contribute to a vineyard's eventual decline in yield and economic viability, including things like soil pathogens, trunk diseases, viruses, water status, and other problems arising from poor management decisions such as soil compaction. Management's decisions made during the establishment or reworking of vineyards can have long-lasting consequences on vineyard health and longevity as well. So in some wine growing regions, it is a common practice for canes to be wrapped tightly around the cordon wire during the establishment of permanent cordon arms. It stands to reason that this wrapping may cause a constriction of the vine's vascular system, becoming worse over time and disrupting the flow of water and nutrients. And as some of you have probably seen, this is especially apparent in the case of older vineyards, where the wire is often visibly embedded within the wood of the cordon, and signs of decline and dead arm symptoms are frequently observed. It's likely that the stress resulting from the constriction of the vine's vasculature could reduce the vine's natural ability to deal with external factors, such as the onset of fungal trunk diseases. And this condition could also be further exacerbated by water and heat stress events, which is becoming more and more of a major concern as these scenarios become more common. So as a bit of a general overview, grapevines, like all other vascular plants, require water to grow and thrive. And the movement of this water throughout the vine, along with essential mineral nutrients, sugars, and amino acids, is achieved via the vascular system. Water is transported in the form of sap, the ascent of which is explained by the cohesion tension theory, whereby the evaporation of water molecules at the leaf surface pulls water from the soil into the roots and through the xylem conduits towards the leaves. Xylem conductivity is determined by the structure and size of vessels as well as their efficiency. It has been demonstrated that xylem morphology is impacted by water availability, and the hydraulic properties of old wood are different than those of young shoots and stems, as vessel length and diameter are actually correlated with stem diameter. And additionally, the ratio between the width of the xylem and the total stem diameter is greater in mature stems. It's been demonstrated that water deficits during the growing season can inhibit vine vegetative growth and photosynthesis, as well as having a negative impact on yield and fruit and wine composition. Such deficits affect the source sink balance of the vine and may therefore affect productivity both in the current growing season as well as in the following years, given that early spring shoot growth is supported by and reliant on reserves. Previous studies have found that the seasonal impacts of water deficit could potentially inhibit starch accumulation, And so it's therefore reasonable to hypothesize that any water stress symptoms caused or exacerbated by the constrictive effects of wrapping developing arms tightly around the cordon wire could lead to a reduction in carbohydrate reserves. And likewise, any reduction in cordon volume occurring as a result of the constriction could directly affect the vine's capacity to overwinter carbohydrates, as the perennial structures of the vine, including the roots, the trunk, and the cordon, are all major storage organs for carbohydrates. And so, in the regions where that tight wrapping method is a common practice, it's popular for several different reasons. Other methods, including placing the canes on top of the wire and securing them in place in 3 or 4 positions, can be more costly and can also be more time consuming by comparison. And so that strategy also normally requires the presence of at least a single foliage wire, as in the absence of any support for developing shoots to latch onto. Arms positioned in that fashion have a tendency to roll under the influence of their own weight, which can disrupt the selection and formation of permanent spur positions and potentially lead to the overexposure of any fruit that may be present. And so wrapping the canes tightly around the cordon wire has the inherent advantage of providing those developing arms with additional support, reducing the risk of rolling and mitigating the need for any foliage wires. It's been reported that constriction applied via plastic straps on olive trees led to a reduction in vegetative growth and canopy volume, as well as a reduction in yield for low vigor cultivars. And that reduction in vegetative growth also corresponded to lower stem water potential, likely as a result of reduced xylem girth growth caused by that plastic strap at that constriction point. A similar reduction in vigor has also been observed after girdling in various orchard trees, as well as in grapevines. And so, based on all of those results that have been seen before, it seems probable that any disruption to the vine's vasculature imposed by wrapping the cordon tightly around the cordon wire could inhibit carbohydrate accumulation in the perennial organs, negatively impacting growth and productivity in the following seasons. Another issue of concern is whether constriction could reduce the vine's ability to deal with the onset of fungal trunk diseases. Such diseases are caused by a range of phytopathogenic fungi and have the capacity to drastically reduce vineyard productivity. Inclusion of the xylem and phloem elements may occur in infected vines, along with wood decay, impairing the translocation of water and nutrients. And so visible symptoms of decline often don't develop until 10 or more years after plantings. And some previous studies have suggested that variations observed in the severity of symptoms from year to year may be more influenced by climatic factors such as rainfall and temperature than by vineyard practices. And so vines may be infected by fungal pathogens without displaying any symptoms at all. And several studies have yielded results indicating that infected vines may be more likely to express symptoms if their health is already compromised by stress. And so, if this is truly the case, then adopting a training method which avoids constriction of the vasculature of the cordon could potentially help to limit the onset of trunk disease symptoms by avoiding a reduction in vine defense response. So the aim of this work that we're going to be looking over just now was to quantify the impacts of different cordon establishment techniques on vine health and longevity. And so we had hypothesized that wrapping developing arms tightly around the cordon wire would result in a constriction of the vascular system, becoming worse over time and potentially disrupting the flow of water and nutrients. And so the main portion of this trial was carried out at 2 small-scale commercial vineyards located in the Barossa Valley, South Australia. And one of those sites featured a 4-year-old planting of Cabernet Sauvignon, while the other featured an older vineyard that was newly reworked. And so that one was a 21-year-old planting of Shiraz. So in both cases, establishing brand new cordons there. So in addition to that main trial, we conducted an additional survey conducted over 2 consecutive growing seasons at 10 other vineyard sites with older cordons ranging in age from 14 to 24 years old and displaying varying degrees of cordon decline. And so this was to investigate the nature of the relationship between cordon strangulation, dieback, and fungal trunk disease symptom expression. So you can see in the images there, particularly in that middle one, some examples of foliar symptoms typical of Eutypa dieback caused by the fungal pathogen Eutypa lata, or Eutypa, as it's more commonly referred to around here. These symptoms comprise stunted shoots with chlorotic and yellow leaves, often cupped and with tattered margins. So each spring, when developing shoots were approximately 50 to 100 centimeters long, the severity of cordon dieback and the presence of Eutypa dieback foliar symptoms were both assessed on 0 to 100% scales. Cordon dieback was assessed as a proportion of the entire cordon length, where the wood had died back and where the canopy was no longer visually present. Foliar symptoms were assessed as a proportion of the remaining canopy only. So the leaf matter that was left over, which displayed those characteristic symptoms that were just discussed a moment ago. And in addition to those measurements, the degree of cordon strangulation was also assessed on a custom 0 to 4 point scale designed by yours truly, according to the visual criteria presented in that table there. And so ranging from no strangulation at all to very severe strangulation. So you can see in this PCA plot that there was a strong correlation observed between the degree of strangulation and the cordon age, which is what one would expect. And sites with younger cordons, such as sites A, B, and C there, are very well separated from the sites featuring older cordons like G and J. So surprisingly, a trend was observed of lower severity of dieback in the cordons displaying the greatest degree of strangulation. And so this was an unexpected result, given that dieback is a condition that is normally associated with advanced cordon age, and especially given that a correlation was observed between degree of strangulation and vine age. And so high cordon dieback also correlated with low plant area index measurements obtained from the Vidi Canopy app. And so this particular app is an app that allows you to take images of the canopy foliage, and it then binarizes those images into a foreground and a background to algorithmically determine certain canopy parameters such as the leaf area, the porosity, and this sort of stuff. And so using this helped to eliminate the possibility of human error during the visual assessments used for the determination of the dieback severity. And you can see how they inversely correlate with each other nicely there. In both years of the survey, a trend was observed of an increase in the visible Eutypa dieback foliar symptoms with an increase in the degree of strangulation, although this was not significant. And so for the main trial at that newly planted Cabernet site and that newly reworked Shiraz site, the treatments included, for starters, a control where the canes selected as permanent arms were wrapped very tightly around the cordon wire in line with that common industry practice. So you can see what that looks like in that image there. Another treatment was placing the cordon on top of the wire. So in this case, the canes selected as permanent cordon arms were placed on top of that wire and then secured in place at 3 or 4 different locations. And then cordons woven through a plastic clip system. So this was a bit of a novel one where those cordon arms were actually woven through that plastic clip system centered between parallel cordon wires. So you can see in that image how the cane was brought over top of that first clip, under that middle clip, and then over top of that final clip. And additionally, at the newly reworked Shiraz site, we included one additional treatment, which consisted of an S bend. So in this case, the canes were wrapped loosely around two parallel cordon wires in a sort of S-shaped bend, as you can see in that figure there. So some of the measurements of vegetative growth we took included pruning weights, as well as cordon circumference and the plant area index using that same app I was discussing a minute ago there. And for certain measurements, we divided the arms into arm sections. So proximal, intermediate, and distal arm sections, beginning from that head or crown area, working their way to the apical point. And you can see what that looks like in that image in the bottom left there, how we divided those arm sections up. So on the newly planted Cabernet Sauvignon site, it appeared that the tightly wrapped treatment reduced the circumferences of the proximal, intermediate, and distal arm sections in the later seasons of the trial. And so these differences in circumference were not observed in the first year of the trial, as you can see in the graphs there, and so seemingly developed over the following growing seasons as a result of those treatments. And so those same tightly wrapped cordons also had lower intermediate and distal pruning weights than the other treatments in the later seasons of the trial as well. On the newly reworked Shiraz site from the second growing season, a trend developed where the cordons which were wrapped around the parallel wires in that S-shaped bend had a significantly lower plant area index than the other treatments on many of the imaging and assessment dates. In fact, including all three assessed dates in that final season. And so while that particular training method extended the cordon, if you can picture it, the furthest spatially into the mid row, so some of those spurs would be facing out sideways from those two parallel cordon wires, logically suggesting that that training method could make those spur positions more prone to mechanical breakage. It actually seemed that in this case, those low planetary index values that we saw were not due to mechanical damage. And so we know that because we also surveyed for this form of damage. And so this was assessed in the weeks immediately following machine harvest on a 0 to 100% scale as a proportion of the total number of spur positions, which seemed to be damaged in a manner that here typical of damage caused by mechanical implements. And so I'll mention that the harvesters used at both sites utilized bow rod shaking systems for the harvest, and one was a towed unit, the other being a self-propelled unit. So the number of spurs lost to mechanical damage increased at both sites each year, as one would expect, as that permanent canopy architecture enlarged. Surprisingly, though, in the final year of the trial at the Shiraz site, the cordons which were wrapped tightly around the wire lost more spurs to mechanical damage than those which were woven through that plastic clip system or trained in that S-bend. And so this result was particularly interesting as some growers purport that method of training, that tight wrapping method, to actually reduce the rate of breakage due to the stability that it provides when justifying its use. But in this case, it seems like that rigidity and that lack of give is actually what caused it to succumb to more damage from those harvesters. And if you picture some of the other training methods that fared better, like that S-bend one, for example, it kind of functions almost like a shock absorber, you know what I mean, in terms of how it could react to that pressure applied from the harvester coming through and give it more give. And it seemed to reduce some of that damage that was succumbed there. So non-structural carbohydrate status was measured from cane samples collected from the different arm sections during winter dormancy, utilizing anthrone and enzymatic assay for that purpose. We could make an argument that if lower concentrations of carbohydrates were observed in canes from the distal sections, so the most extreme sections of the cordon arms during dormancy, then the training method may have been beneficial in allowing the movement of the carbohydrates from those distal canes to the perennial structures of the vine for overwintering. And so in this case, those transiting carbohydrates would have to move through the conduits of the cordon arms, where the vascular morphology could be impacted by the constrictive pressure applied from the different training methods. When considering the carbohydrate levels of the different cordon arms in this trial, however, one common observation was that if the levels of sugars, starch, and total non-structural carbohydrates were low in one particular arm section, they were likely to be similarly low in the other arm sections as well, suggesting that the treatments did not have much of an impact in terms of restricting that translocation of the carbohydrates along the cordon arms. 1-year-old cane samples were also collected from the distal arm sections during dormancy for the purpose of assessing parameters of vascular morphology, such as vessel diameter and cross-sectional vessel density with the use of a stereo microscope. And additionally, cane samples were also assessed by micro-CT for metrics including xylem conduit volume, vessel thickness, connectivity density, and theoretical specific hydraulic conductivity. And so, while no conclusive results were obtained from the analysis of those 1-year-old cane samples, luckily, as a final investigation in the trial, when all of the other measurements were already complete, it was possible, with the permission of our cooperating growers, to destructively remove entire cordon arms for the generation of 3D models and their analysis. So this really brought the whole project together and allowed for a direct assessment of the impact of the different treatments on the local morphology of the cordon xylem. And so you can see in that graphic there how after only 4 growing seasons, the tightly wrapped cordons had a clearly distorted shape relative to the other treatments. And it's very apparent where the wire was beginning to become embedded within the wood of the cordon. So on the 4-year-old Cabernet, the volume of the xylem conduits of the tightly wrapped cordons were significantly lower than the other treatments relative to the total cordon volume. And so those are expressed as a percentage of that volume of the xylem conduit relative to the entire cordon volume. The volumes of the xylem conduits of the cordons which were placed on top of the wire were the greatest out of all the treatments, which makes sense when you consider that they were the least constrictive training method out of all the treatments. And you can see on the right there how the results on the 21-year-old Shiraz followed that same trend as what we saw in the Cabernet, although not significantly. You can also see how those S-band ones seem to fare pretty well as well in terms of their xylem conduit volume. The cordons which were wrapped in the S-bend had significantly fewer average vessels per unit of cross-sectional area than the other treatments. And there's no obvious reason for why that was the case, but the shape of the cordon imparted by that particular training method did have the greatest deviation from a straight structure, if you picture how it was wrapped loosely around those parallel wires. And for the Cabernet, that lack of a difference between the number of vessels suggests that it was actually the size of the vessels rather than their total number, which had a greater impact on the total xylem conduit volume. And so that was further supported by measurements of individual vessel thickness, which was acquired with a sphere fitting algorithm during the 3D analysis. So a very nice, robust way to take this sort of measurements, which essentially equates to the vessel diameter. And so the cordons which were wrapped tightly around the cordon wire had significantly thinner vessels than those that were placed on top of the wire or woven through the plastic clip system, as you can see on the left there. The tightly wrapped cordons also had a significantly lower number of connections between vessel structures, which is important as these connections are a vital element of the sap movement pathway. And this suggests that these cordons could already be less efficient and hydraulically integrated even after just the few year span of the trial. Theoretical specific hydraulic conductivity was calculated to allow for an assessment of the impact of the treatments on water transport. And so the Ks values of the tightly wrapped 4-year-old Cabernet were lower than the other two treatments, suggesting a likely impairment in xylem transport efficiency. And these results were highly significant with a p-value of less than 0.0001, with the conductivity values for the tightly wrapped cordons actually being 50.2% lower than the cordons which were woven through the clip system, and 57.2% lower than the cordons trained on top of the wire. And so several results suggest that the constriction applied by the tight wrapping had a negative impact on vegetative growth. And these included those reduced circumferences of the arm sections of the tightly wrapped cordons in the later seasons of the trial, as well as lower pruning weights. Cordon xylem morphology measurements also suggest that their capacity for normal hydraulic function was compromised. And it's likely that the differences observed between the applied treatments would continue to become more pronounced with further observation, as cordon constriction is a condition that becomes progressively worse over time. And if using the visual scale presented earlier from the disease survey, for example, none of the cordons that were assessed for these purposes would have been rated greater than a 2 out of 4 in terms of their degree of strangulation. So all said and done, you could expect that those negative outcomes we were looking at could be much more pronounced in older and more strangled cordons. So by better understanding the potential benefits of adopting cordon establishment techniques which avoid a constriction of the vine's vasculature, vineyard managers may now hopefully be provided with a strategy aimed at improving vine health and longevity. And while there are certain benefits associated with wrapping developing cordon arms tightly around the cordon wire, avoiding a constriction of the vascular system may be a reason of great enough concern to reconsider that practice, as the results of this trial have indicated. So, as another part of this work, we looked at the benefits of adjusting the length of canes selected as permanent cordon arms based on an assessment of their apparent vigor. This was to investigate a potential approach for solving the problem of where if canes selected as permanent cordon arms are very weak and small in diameter, they may lack the vigor needed to support uniform bud burst and the growth of new shoots. The impact of this is especially apparent for nodes or buds in the center of low vigor arms, as grapevine shoots display apical dominance, as well as having an acrotonic tendency to prioritize branching in that shoot distal zone. And so the decision of whether the length adjustment was warranted was based on a visual assessment, taking into account cane length and diameter, as well as node number. And so low vigor canes were cut back in length with the number of nodes retained based on the apparent vigor of the cane, as can be seen on that left arm in that image. And canes which were deemed to have sufficient vigor to support uniform growth were left at full length, as can be seen on that right arm. And the hope was that by adjusting the length of the canes and limiting the number of nodes retained, successful bud burst would be encouraged, as well as the growth and development of shoots of greater diameter and better suited for selection as permanent spur positions. Canes that received a length adjustment were then brought to full length during the course of the growing season by selecting an ideally downward facing shoot from the distal portion of the cane near the cutting point, and then training it horizontally along the cordon wire. And so while this method required some attention to secure those extension as they grew to full length, the desired outcome was again healthier, more productive cordons. So after the first growing season, the intermediate or middle arm sections of cordons, which had undergone that length adjustment, had significantly higher pruning weights than the control cordons when considered on a per meter basis. No difference was observed between the pruning weights of the intermediate arm sections in any following year, or between the other arm sections, or on a whole vine basis in any year. After the first growing season, the intermediate arm sections of cordons which received the length adjustment had significantly more canes per meter than control cordons, and the average weight of those canes was also higher. After the second growing season, significantly less canes were observed in the distal portion of the length adjusted canes. And so this is logical when you consider that at that time, the distal portion of newly extended arms did not yet have fully formed spur positions. And so their fruit bearing canes originated directly from the nodes of the extended cordon rather than spurs. And so therefore typically had just one cane per spur position rather than two canes per spur position. No differences were observed in plant area index between length adjusted and control fines during the first growing season, but length adjusted cordons had significantly lower plant area index value than control cordons on two imaging dates in the second growing season, one date in the third season and two dates in the fourth season.
Sorry about
No
the,
difference.
I'm sorry about those blue lines. Just continue.
Problem.
Yeah.
No difference was observed between the circumference of the proximal, intermediate, or distal arms sections of cordons which received that length adjustment and control cordons in any year. And so this actually includes the second winter, where one might have reasonably expected that the distal arm sections of adjusted cordons might be smaller circumference than the control cordons, as at that time they were comprised strictly of 2-year-old wood rather than the 3-year-old wood, as was the case with most of the control cordons. And so the fact that no difference was observed in cordon circumference suggests that the delayed development of the distal arm section inherent to that method did not have a negative impact on perennial wood structure. And so that's a very encouraging result that could actually make some practitioners more comfortable with adopting the use of that method. And while the results of the trial did not indicate a long-term beneficial response as a consequence of the length adjustment, this doesn't mean that the vines did not benefit from the practice, as during that first growing season, the treatment was seemingly successful in encouraging vegetative growth, especially in the middle of the cordon arms. And again, that's a critical point, as in situations where there is insufficient vigor for unhindered bud burst and shoot development, that effect is normally the most pronounced in that middle arm section. While the length adjustment did seem to encourage the growth of canes in the middle of the cordons that were both bigger and more numerous, the benefits of that may have been limited to a single growing season. It's possible that this treatment could have a stronger beneficial response in other cultivars, particularly those in low vigor environments, which struggle with apical dominance. And so further research focusing on such an environment where the control vines are more likely to suffer from severe detrimental effects arising from a high retained known number could better illustrate the comparative beneficial response of that treatment over a longer term. So I just wanted to make a few acknowledgments there. This work was supported by the University of Adelaide as well as Wine Australia, with levies from Australia's grape growers and winemakers and matching funds from the Australian government. And I also just wanted to express gratitude to the growers who allowed us access to their vineyards for the conduction of the study, as well as Craig Johns and Simonette and Search for their guidance on trial establishment. So there's my contact info there. Feel free to reach out to me at any time with any questions. I believe this is normally the point, Aaron, where we would stop the recording and open it up to live questions.
Okay. We'll do that now. Thank you.